Hi everyone, my name is Rex, and I'm glad to present you a paper called Graph Neural Networks for Multimodal Single-Cell Data Integrations. And it's a accepted paper in the conference KDD 2020 by researchers in Michigan State University. And something about myself, I'm a PhD student at the University of Toronto. My research focus in machine learning in healthcare and biology domain, basically try to solve all the problems that using machine learning or deep learning, to be more specific. Okay, some backgrounds about this paper. So with the technologies, well, some caveat first, you need to have some basic understanding of what single cell data is. And also you are expected to know some basic knowledge about graph neural networks. Okay, with that being said, the backgrounds, with the advancements of single cell technology, we can simultaneously measure multiple single cell features now, but it is still challenging to effectively utilize the complementary informations from multimodal data. And NeurIPS has a challenge. Well, it's now hosting year by year on single cell data integrations. And this paper is actually the winner team for last year's challenges subtasks. The whole challenges have 3 different subtasks, and this paper was the winning team for the first subtask, which I will talk about what the subtask is and explain more about this paper. So after they winning this challenges, they extend their methods to work with other 2 subtasks, and now they are the best performing models for the overall challenges. Okay, so what are the 3 different subtasks? The first subtask is modality predictions. Basically, it's given, okay, let me open the pointers. Basically, it's given 2 different modalities. We're trying to learn a mapping function that map one modality to the others. And the evaluation matrix, it's the RMSE score. Basically, that's the difference between the predicted value and the ground truth value. And this paper was the winners of this task. And the second task is called modality matching. Also, it's giving 2 different modalities. We're trying to learn a way to map the 2 modalities. To be more specific, so let's say we have the modalities of RNA and the taxid. We are trying to map the cells across different 2 modalities. Right, the cell 1 in RNA is the cell 3 in the taxid, something like that. And the output of this network is a similarity matrix. Well, with the probability, with each element is the probability of the cell I are paired with the cell J in the 2 modalities. And the final matrix is calculated by the average of the ground truth pairs. So here we can see that as the average of 0.4 with 0.5 and 0.8. And the third subtask is to get the joint embedding. So the goal of this task is to, given 2 different modalities, you will need to learn a joint representation of the 2 modalities for each cell. And then the embedding will be evaluated on a lot more metrics, including like batch corrections, label preservations, and even the self-cycle preservation. They offer lots of metrics for evaluating this. Okay, so these are all the 3 different subtasks. And for the next sections, I will first go over the general framework, the general GNN framework proposed by this paper. And then I will talk about what are the modifications they did for each different subtask. Okay, so for the general GNN framework, I think this is the most novel part about this paper. So how they construct so for GNN network, we need the adjacency matrix, and also we need the node embedding, right? So now let's deal with the adjacency matrix first. How they construct the adjacency matrix is they not only consider the cell-to-cell matrix, but also the cell-to-feature matrix as well. Basically it's a bipartitional graph where you have the cell nodes with the feature nodes, but the cell nodes are all zeros. The elements of the cell nodes are all zero. Feature nodes to feature nodes, it's all zero as well. Well, they can add some extra prior knowledge here, but we will talk about that later. So let's just assume that here are all zero. And then in terms of the cell nodes and the feature node, they just put their modality inputs, whatever the raw data here, they just put it here as the connection, as the weighted connection edge, as the weighted edge of that adjacency matrix. Well, of course, here it will be just the transpose of this part. All right. So to summarize, the whole dimension is n plus k, and with the top left and the bottom right are all zero. Okay, now we have the adjacency matrix. The next thing is the getting the node embedding. So I think this is the part that this paper is not clear about. For the cell and feature embedding, they just said that, okay, we can initialize it to different dimensions like d prime and then d double prime. That based on that initialization can be depending on the task specifics. And then they also give examples. For example, that the feature embeddings can be just a one hot encoding of which feature dimension of that feature belongs to. For example, you can just be AKA just an identity matrix of the k dimensions. And also for cell features, that's okay. That's very confusing. They just claim that you can just put a one-dimensional zero as the features for all the cells. And also for different subtasks, they didn't explain how they do the initialization for the subtask. So overall, this part is really not clear. Okay, so now we have the node embedding with the adjacency matrix. For GNN, we need the aggregations, right? Or the updates within different layers. So here is the update functions. So we have the embedding of the node I at layer L. And in each iterations or at each layers, we will do the aggregations of the embedding from neighboring nodes. And then using the we put the aggregation informations together with the embedding from previous layer, and then use it as the updates for the embedding for the next layers. So put it into a mathematical way. For example, here in layer 1, basically we just get the weight matrix and the bias matrix to go through the features, and then pass it through activation functions, and then we get the first layer embedding. Okay, so as you can imagine, the different thing here with other GNN papers is that we now have two types of nodes. We have the cell nodes with the feature nodes, right? So this paper proposed that we should do different aggregations for different type of nodes. Okay, let's denote the V as the sets of feature nodes and U as the sets of cell nodes. So in one sentence to summarize all of this is that they do different aggregations. So here, for example, we can do cell nodes from feature nodes. When we do cell nodes from feature nodes, we have specific weight matrix, bias vectors for each layer of the cell node to feature node. For feature node to cell node aggregations, we also have different bias vector and weight matrices. Because it's a bipartitional graph, right? And only the cell nodes are connected with the feature nodes, not within a cell. Okay, so finally, it will be task-specific heads. So what they will do is they take the node embedding for each layers, and then they figure out some way to concatenate all of them. It can be functions, or it can just be simple concatenations. And then we have the heads, whatever is trying to predict using that concatenated embeddings. Okay, this part is easy to understand. Okay, next, I will talk about how they do the modification for different subtasks. So for the first subtask, they do two different modifications. The one is leverage the pathway data, and the other one is the weighted aggregation functions. So the first point is, how do they utilize the pathway data? It's by adding feature to features adjacency matrix. So we all know that for pathway, each pathway can have like 50 genes, right? So within the same pathway, the author treat them as we can believe that the genes are correlated. So here they leverage that informations. If two genes are within the same pathway, they treat it as a connected nodes. And in terms of the values, they calculate it by doing the cosine similarity between the raw features. Okay, so by doing this, now we have the P matrix that connect the feature node to feature nodes. And the second thing is the weighted aggregation functions. So since, so this part only make changes to the feature nodes, because the feature node now not only connected with the cell nodes, but also with other feature nodes. Here, we will have another weighted, we'll have another aggregation function for feature node to feature nodes. And then they use alpha as a weighted parameters to have a weighted sum of the aggregation between these two. Okay, and here's the results of the task one. So there's lots of state-of-the-arts, what the previous methods that they compares, and then here are the results. Even though they sort of achieved the overall best performance, but it's not at a substantial differences with others. But overall, it's good. Okay, and now regarding the modification to task 2, here I outline them in steps. So first, they construct. So now let's say now input is two different modalities. We first construct separate cell feature graph for each modalities. And then we do the GNN training, right? The GNN trainings can give you the final embeddings for each modalities. So now let's say we have the embeddings for all the cells within each modalities. So they have two different laws for training the learning of the embeddings. The first laws being the matching laws. So they first calculate the cosine similarity of the embeddings from the two modalities. And then using that cosine similarity, they do a softmax. Oh, that softmax pretty much give you what's the probability of this cell mapped to the cell I mapped to the cell J in the other modalities. And we also have the ground truth of whether they are actually mapping. So the actual loss will be sort of like cross entropy loss, right? Okay, that's the matching loss. And then they also do some tricks, for example, like a reconstructions loss and a prediction loss. So here you can see that they also train a prediction hat. Well, they just the two-layer MLP. They just train a two-layer MLP that can turn the embeddings from modality 1 to the raw features of modality 2. That's the predictions. Well, vice versa for modality 2 to the raw feature of modality 1. Also, the same mapping function will be used to reconstruct the modality 1 embeddings to modality 1 raw features, and also modality 2 features to modality 2 raw features. So this is the 4 different laws that added up to the matching laws for the task 1. It's a lot of laws, right? Yeah. Okay, and the result for task 2 is that well, they overall achieved the best performance. As you can imagine, as you can remember, it's the weighted average of the probability, is the weighted sum of the average probability in that giant similarity matrix. So the numbers can be really small. And the baseline are all close to 0. And for their methods, they achieve like a best performing, well, now we can see a significant improvements of this method regarding the others. And regarding the third task, which is to learn the joint embedding, they first do a latent semantic indexing. Apparently, this is dimensional reductions way to get a lower dimensional embedding first. So they use this. They use LSI to pre-process the two modalities and then concat them to get the cell features, to get the features. And then they construct only one cell feature graph using that concatenated features. And then we also get the GNN embeddings from there, right? And then using that embedding, we train a prediction head just to reconstruct the raw concatenated features. But also, we also leverage the cell type informations. So we also using that prediction head to predict whether they belong to the correct cell type, and then use the cross entropy loss here. And lastly, they add another regularization term in the end for the loss. So that's the loss for task 3. And then here shows the performance of the task 3. Now you can see that they achieved the best performance from 4 out of 7. Sort of like a good, right? It's not too bad. Okay, so finally we can reach discussions here. Here's my thinking about their pros and cons. So regarding the pros, I think the cell feature graph is definitely a novel idea. It's the first time I see this kind of ideas that applying in single cell studies. And the second one is the framework. It's very flexible. It can be adapted to different subtasks with good performance. But also, I think the coin is the modifications of each subtask is not trivial. They seem to add lots of tricks to make it work, like the fancy, like the cup of loss that you just see, right? And also, I think the whole challenge, the NeurIPS challenge, is focused on integrations within two modalities, from predictions, from the matchings, and the joint embedding. They are all about the two modalities. So I think the framework is not scalable to multimodality yet. Because all the cell, as you can see when they construct the adjacency matrix, all the features are in the featured nodes, and then the dimension can be super large, right? So I think the authors can improve on this point. Okay, so that's the end of the presentations, and then I hope you enjoy reading this. If you have any questions, feel free to leave a comment, and we can chat below. Thanks.